The internal world of a eukaryotic cell is a realm of breathtaking complexity, an urban landscape of organelles and molecular machinery operating with a precision that rivals the most sophisticated human engineering. Central to this cellular metropolis is a logistics and transport system of unparalleled elegance, responsible for moving vital cargo proteins, lipids, and signaling molecules from their sites of manufacture to their designated destinations. This cargo is packaged into small, membrane-bound sacs called vesicles, which barred from one compartment and fuse with another in a ceaseless, highly regulated flow. For decades, the fundamental principles governing this traffic remained an enigma, a black box at the heart of cell biology. The 2013 Nobel Prize in Physiology or Medicine, awarded to James Edward Rothman, Randy Wayne Schekman, and Thomas Christian Südhof, celebrated the definitive unraveling of this mystery. Through three distinct yet convergent lines of inquiry, these scientists dissected the molecular machinery that controls vesicle transport, revealing a universal mechanism of exquisite specificity and temporal control. Their collective achievement provided a coherent molecular framework for a process fundamental to life, transforming our understanding of everything from hormone secretion to the firing of a single neuron. The intellectual groundwork for this discovery was laid decades earlier, most notably by the pioneering work of George Palade, using the powerful combination of electron microscopy and pulse radio labeling. Palade charted the course of newly synthesized proteins, revealing what became known as the secretory pathway. He visualized proteins moving from the endoplasmic reticulum through the stacked cystinae of the Golgi apparatus, and finally into vesicles that ferry them to the cell surface for release. Palade's work provided the definitive anatomical map of this cellular highway system, a discovery that earned him a share of the Nobel Prize in 1974. His elegant studies established the what and the where of secretion, yet the how remained elusive. The identities of the molecular motors, the traffic controllers, and the docking machinery were completely unknown. It was this profound question of mechanism that Randy Schekman set out to answer. Employing the formidable power of genetics, Schekman made the insightful choice to work with a simple yet powerful model organism, the budding yeast Saccharomyces cerevisiae. He reasoned that a process as fundamental as secretion would be conserved throughout eukaryotic evolution, and that the genetic tractability of yeast offered a direct path to identifying the essential components. His experimental strategy was a master stroke of design. He generated a collection of yeast mutants that were temperature sensitive. At a permissive temperature, these cells grew and secreted proteins normally. When shifted to a higher, non-permissive temperature, a specific genetic defect would manifest, causing the secretory pathway to grind to a halt. The genius of this approach lay in its consequent proteins destined for secretion would accumulate at the specific step blocked by the mutation. By observing where these proteins piled up using electron microscopy, Schekman could effectively create a living, dynamic map of the pathway's functional stages. This arduous screen identified a suite of genes he named Sec for secretory. The mutants fell into distinct categories based on their morphology. For instance, Sec1 mutants accumulated vesicles at a plasma membrane, unable to complete the final fusion step, while Sec18 mutants showed a massive pileup of vesicles between the endoplasmic reticulum and the Golgi. I categorizing these mutants class showing cytosolic accumulation, class B in the endoplasmic reticulum, class C in Golgi-derived vesicles to order the function of the SEC genes chronologically, providing the first a parts list for the secretion machine and a functional blueprint for how those parts were assembled into a coherent par. While Schekman was meticulously identifying the genetic blueprint in yeast, James Rothman was tackling the same problem from a biochemical perspective. His ambition was to do for cell biology what had been done for metabolic pathways to deconstruct a complex cellular process into its constituent parts and reconstitute it in the pristine, controlled environment of a test tube. He focused on a single, discrete step in the secretory pathway, the transport of proteins from one Golgi cisterna to the next. The cell-free assay he developed was a model of scientific elegance. He prepared Golgi membranes from two different cultured mammalian cell lines. The donor membranes were harvested from cells infected with vesicular stomatitis virus, which produced a specific viral glycoprotein, known as the SVG. Crucially, these cells were genetically deficient, so their version of the SVG lacked a particular sugar molecule, N-acetylglucosamine. The acceptor membranes were from normal, uninfected cells. Transport was deemed successful only if vesicles carrying the VSCG protein budded from the donor Golgi and fused with the acceptor Golgi, where resident enzymes would add the missing sugar. By using a radioactive form of N-acetylglucosamine, Rothman could precisely and quantitatively measure the efficiency of this transport event. This robust assay became his workhorse. By systematically fractionating the cellular cytosol and adding components back to the assay, he could purify the proteins essential for transport. This led to his first major discovery, a protein he named NSF, phenethylamide sensitive factor, because its activity could be blocked by this simple chemical. He subsequently identified another set of proteins, SNAP, for soluble NSF attachment proteins, which were required for NSF to bind to the Golgi membranes. Rothman had isolated the core components of the intercompartmental transport engine. The general machinery for vesicle budding and fusion was now coming into sharp focus through the parallel efforts of Schekman and Rothman. However, many biological processes demand a level of regulation far exceeding the steady, constitutive flow of the general secretory pathway. Nowhere is this more apparent than in the nervous system. At the synapse, the junction between two neurons, communication occurs when an electrical signal, the action potential, triggers the near-instantaneous release of neurotransmitters. These sickling molecules are stored in synaptic vesicles, which must fuse with the presynaptic membrane within milliseconds of the signal's arrival. The trigger for this rapid fusion is a sudden influx of calcium ions into the nerve terminal. The problem of how this calcium signal was coupled to the vesicle fusion machinery became the central focus of Thomas Südhof's research. He set out to identify the molecular sensor that could detect the rise in calcium and translate it into the physical act of membrane merger. Through a systematic search for calcium-binding proteins in synaptic vesicles, his laboratory identified and characterized a protein named synaptotykmin. He demonstrated that synaptotykmin was the principal calcium sensor. It contains domains that bind multiple calcium ions, and this binding event induces a conformational change that allows it to interact with other proteins on both the vesicle and the target membrane, thereby triggering fusion with the necessary speed and precision. Südhof's work provided the critical regulatory link, the molecular switch that gives the nervous system its unparalleled temporal acuity. The convergence of these three research programs, Schekman's genetics, Rothman's biochemistry, and Südhof's neurophysiology, led to a grand unifying theory. Südhof's work in neurons identified the specific proteins at the synapse responsible for the final fusion event. He found a protein on the synaptic vesicle, synaptobrovin, also called VAMP, and two proteins on the target plasma membrane, syntaxin, and a protein known as SNAP25, which, despite its name, is unrelated to Rothman's SNAP. He showed that these three proteins formed an exceptionally stable complex. Simultaneously, Rothman's laboratory found that his purified NSF and SNAP could bind to a membrane receptor complex that, remarkably, contained these very same proteins identified by Südhof. The pieces of the puzzle clicked into place. Rothman proposed the elegant and powerful snare hypothesis to explain the specificity and mechanics of membrane fusion. The hypothesis posited that vesicles carry specific protein markers, which he termed V-snare for vesicle snare like synaptobrovin. These V-snare would only pair with a coordinate set of proteins on the correct target membrane, the T-snare for target snare like syntaxin and SNAP25. The formation of this trans-snare complex, a coiled coil bundle of 4 alpha helices, acts like a molecular zipper. It pulls the two membranes into intimate opposition, overcoming the substantial energetic barrier to lipid bilayer merger and driving fusion. Following this event, Rothman's NSF and SNAP proteins would act as a disassembly machine, using the energy of ATP hydrolysis to pry apart the extremely stable snare complex, thereby recycling the components for subsequent rounds of trans. The snare hypothesis provided a stunningly coherent framework that accommodated the findings from all three laureates and explained a fundamental aspect of cellular organization. It offered a universal mechanism for ensuring that vesicles deliver their cargo to the correct address, a principle that operates in all eukaryotic cells, from yeast to human neurons. The impact was immediate and profound, opening up entire new avenues of research. The hypothesis provided a testable model that has since been refined and elaborated upon, but its core tenets have stood the test of time. Its medical relevance became starkly clear with the realization that the potent neurotoxins produced by the bacteria responsible for tetanus and botulism are, in fact, highly specific proteases. These toxins cleave the snare protein's tetanus toxin cuts synaptobrovin, while botulinum toxins target syntaxin, SNAP25, or synaptobrovin by paralyzing synaptic transmission and causing the devastating clinical symptoms of these diseases. This discovery provided a direct molecular explanation for the pathology of these ancient afflictions. Furthermore, subtle defects in the machinery of vesicle transport are now implicated in a wide spectrum of human disorders, including diabetes, immunological deficiencies, and various neurological and psychiatric conditions. The initial hypothesis, while powerful, has been enriched by subsequent discoveries. Researchers have identified a host of regulatory proteins, such as the MUNC18 and complexin families, that modulate SLARE complex assembly and function, adding layers of sophisticated control that fine-tune the fusion process, particularly at the high-speed crucible of the SYN. In conclusion, the collective discoveries of Rothman, Schekman, and Südhof represent a triumph of modern biological inquiry, a testament to the power of combining diverse experimental approaches to deconstruct a complex problem. Schekman provided the genetic parts list and functional order of the secretory pathway. Rothman isolated the core protein machinery and reconstituted its function in a cell-free system, revealing the biochemical engine of fusion. Südhof uncovered the precise timing mechanism, explaining how the universal machine could be adapted for the split-second demands of neuronal communication. Their combined work illuminated one of the cell's most fundamental processes, transforming vesicle transport from a hazy concept observed in static electron micrographs into a dynamic, molecular-defined choreography. This new understanding has become a cornerstone of modern cell biology, providing the intellectual foundation for countless studies into health and disease. The graceful dance of vesicles, once a deep cellular secret, was revealed to be governed by an elegant molecular logic, a symphony of protein interactions whose fundamental principles continue to inform and inspire scientific discovery across the globe.